Welcome, everybody. My name is Joanne Allen. I'm with the Fairfax County Urban Forest Management Division, and I'm the Forest Pest Branch Chief for my group. And today we have a very exciting webinar featuring the newest pest to our area, beech leaf disease, which is something of grave concern for all of us in the green industry, and for those living in the area, just because beech is such a dominant tree species. So we have a great panel of folks here with us. And before we get started, I wanted to just cover some housekeeping. First, everyone, all the attendees are muted, so unfortunately you won't be able to, unmute yourself and chime in with any questions. But if you do have questions, take a look at the bottom of your screen. In between participants and chat, you're going to see an icon with two little word bubbles that says Q&A. So use the Q&A function if you have any questions, and we have staff behind the scenes that will be helping answering those questions. If there are any questions that I feel that would be great for the speakers to address, we'll be doing that at the end of the webinar. So we discourage the use of the chat, not really necessary. If you just have questions, go ahead, use the Q&A. The chat isn't monitored. So we will not, if you post a question on the chat, we're not going to answer it. So just to review the agenda for today, we have speakers from Fairfax County Urban Forest Management. We have Catherine Layton. We have Danielle Martin from the USDA Forest Service, Brady Holko from Davie Tree Lab, Dr. Sarah Tangren from Metropolitan Council of Governments. So we have our first speaker, Catherine Layton, if you'd like to get started.
Hi, everybody. As Joanne said, I am Catherine Layton, and I am one in a team of urban foresters in the Fairfax County Urban Forest Management Division. And along with other urban foresters in forest conservation and ecologists in the Park Authority, we work to care for and increase the tree canopy in the county. Well, I'm going to be introducing the subject. So let's start with introducing the American beech. The American beech is a very common tree. In Fairfax County, we have about 9% of our tree cover is beech. It's a beautiful tree. It has a very nice shape and beautiful fall colors as well. It does cover about a third of the United States on the east, all the way from southern Canada down to Georgia. And one of its features is its thin gray bark. And unfortunately, it's often been used as a background for graffiti. And that can be harmful to the tree. Please don't do this, but this is one way you know you're looking at a beech. Healthy American beech leaves are elliptical, and they have serrated edges, and they are usually just very uniform. And healthy American beech saplings, in the picture on the right, you see that they retain their leaves. Some of the younger, more mature beech do that as well, mostly in the lower part of the canopy. But that's another way you can tell that it's a beech. They will hold on to those leaves all the way till spring, when the buds are ready to pop. And soon as that pops out the leaf, those little leaves will fall down. And it adds to the mulch and nutrients in the forest floor at a time when other trees are not dropping. So The diseased American beech foliage typically is recognized first when there's the first year or two, you may see this typical striping in the leaves. It's in between the veins, and it can start with just a little twig that has it. You may not even notice it for a couple of years because it will build up and there'll be more branches that have it. As it spreads, and the leaves will become thicker and they'll be leathery and curly, and more of the leaves will be affected. That bottom photograph shows how they can become leathery and curled up. Also, the leaf buds, before they open, they will turn dark and become very dry and brittle. There are some other diseases that might be confused with them, but they really don't look exactly like it. When you're looking for beech leaf disease, we recommend looking in the beginning of the summer. So right now would be a good time and on into June. But after that, some of these other normal diseases and, little aphids will start working on them, and you'll have more and more of that as the summer progresses. The woolly beech aphid rolls itself up in a leaf. It's a sucking insect. There's a mite and an anthracnose, just more of a fungus. I put in parentheses there the boogie woogie aphid. That one I recommend. If you've never seen boogie woogie aphid, look it up on YouTube and watch them. They have a very interesting behavior, which gives them their name, boogie woogie, and you might enjoy looking at that. So as time goes by, more and more leaves are affected and fewer leaves are produced. On the photo on the right that's at Berkeley Park, you can see how many more of the leaves have turned dark and have curled up. The trees will decline as fewer and fewer leaves are there to provide the nutrients they need. And the seedlings and saplings can die in only 1 to 3 years. Mature trees can die in 4 to 10 years. Fortunately, there has been observed a certain percentage, and this is like really ballpark. It could be anywhere from 1 to possibly up to 5% of the trees in a forest may be resistant. And I use the word resistant loosely because it just means that they may survive the whole forest decline and appear to be unaffected. And that's a good thing. We can actually work with that to produce some beech that might make it later on. Beech trees are very important to wildlife. They're very full of fat and nutrients, and a wide variety of wildlife eat them. Everything from bear to birds to deer, turkeys, and waterfowl. They are tied with red maple for the most common tree species in the county. And as I said, that's about 9% of the urban forest. That is really significant. And we expect that as beech leaf disease spreads through the county, it's going to make a lot of difference in our forested areas. So here's a map that shows the spread of beech leaf disease, and it started in 2012 up by Lake Erie. And you can see how incrementally, year by year, it just spread and spread. And it's an extremely quick spreading disease. And I'm not going to go into more details on this because we have researchers who are going to talk about this. But just to note that the spongy moths were first accidentally released in Massachusetts in 1869 and took over 100 years to reach us in Fairfax. This is just an amazing spread. So we did a little bit of looking at this nematode. I'm going to introduce the nematode to you because this is what's been associated with the disease. Every disease has a source. At first, people didn't know what it was. A lot of research was done, and it turns out that there's a nematode, which is a roundworm, that is causing the disease. And there'll be a lot more information given to you about this. I just wanted you to see that we did photograph one in our lab. It's a little curly things, if you see those. That's all it is. And there are thousands of different nematodes that inhabit every niche and ecosystem on the earth. Each kind is specifically adapted to its host. Nematodes have been identified from fossils dating way back to 40,000 years ago. They can be beneficial or detrimental to specific plants because they are so specific to their hosts, and they're not all bad. Some nematodes are used to protect important agricultural crops or control mole crickets on golf courses. So they do have their good points. There are so many of these that if you can imagine this, if you can imagine a picture of the earth with all the living things highlighted, and then imagine that you had Photoshop that could eliminate everything except the nematodes. If you did that, you would be left with this amazing photograph of like a skeletal map of everything alive on the planet in front of you. I thought that was pretty cool. So here in Fairfax County, we're doing a lot of monitoring. We don't have anything that we can use for combating it yet for forested areas. However, it's good for us to know where it is. So we last year we identified it in areas where we knew there were beech stands that were identified by the park authority, and beech stands identified by our field staff from our own field work. And then we delineated the BLD infestations that we found, estimating how many acres they are. We established 4 study plots as part of long-term BLD study by the USDA Forest Service that Danny will probably talk about in a few minutes because we're working with her on that. And then this year, we're going to do 200 countywide surveys looking for beech leaf disease and respond to any reports that we get of it. Continue the long-term study with the U.S. Forest Service and delineate any new infestations. As far as what we're going to do for treatment, if we can find some, we rely on an integrated pest management system. And this is something that the EPA defines as that first paragraph up there. It's environmentally sensitive and it relies on a combination of common sense practices. IPM programs use current and comprehensive information on the life cycles of the pests and their interaction with the environment. And that's important to remember because with beech leaf disease, there is amazing research going on now to do just that. Figure out everything about the pests or the organism that can be found out, because that is the way that you can figure out ways that might work to control it. So some of the more common pests that you see right there, you see the spongy moth, used to be called gypsy moth, also hemlock woolly adelgid, and the emerald ash borer, and our more recent neighbor, the spotted lanternfly. These are examples of where we use a variety of these things over on the left to figure out what to do with them. First, you figure out, what is an acceptable pest levels? We're not going to be able to exterminate all of these. They're going to make their way and just become a part of our land here. What we do need to do is just keep them at an acceptable pest level. So that's something that remains to be determined with beech leaf disease. An example of cultural practices would be for spotted lanternfly, get rid of the tree of heaven. They attract them a lot. If you don't want them on your property, you can get rid of those, and maybe your spotted lanternfly populations will remain low. Monitoring is very important, or you don't know what you're dealing with or what you're doing. An example of mechanical controls would be for spongy moth, putting up burlap and actually removing them and putting them in soapy water. They're biological controls. That is using things like stingless wasps, or perhaps beetles, anything that came from where the bug originated that might be able to be used here. But that's very tricky. It has to go through a long process to experiment with it, to test it, to be approved by the EPA. And yet, when some of these that have gone through this process have been very useful. And then responsible use of synthetic pesticides. That's always a part of IPM as needed, but I emphasize responsible and following a label and only using it as needed. And I'm going to add one more here, and that would be one we touched on before, is the possibility of trees being resistant, and using breeding programs to encourage them to reproduce. If you'd like to report sightings, you can do so on iNaturalist. And if you have questions, please contact us at pestmail at Fairfax County. And there's QR code if you would like to use that for iNaturalist. And here is our Urban Forest Management Division phone number, and again, our pest mail address if you would like to contact us. And that's all I have in terms of introduction for you. I'm going to turn it over to Danny, who's going to talk to everybody about her research with the Forest Service.
So I'm Danielle Martin. I work for the Forest Service. I'm a forest pathologist. I've been in the Morgantown field office for 13 years. I've been working on beech leaf disease, I think, for about 8. So I was going to talk a little bit about some of the projects I'm working on, and mostly about transmission and spread mechanisms. So just a really brief introduction. I know that Catherine already did an intro, but I don't know how to start without doing an intro, so I'm going to do one anyways. So beech leaf disease is a novel disease found to affect American beech, European beech, and Oriental beech, the latter being mostly ornamental species in our country. To date, no beech in affected areas have been shown signs of resistance. However, some have shown tolerance by maintaining similar levels of infection for several years and not declining as quickly as nearby trees. Beech leaf disease symptoms have been shown to be associated with the nematode Litholanthus crenata. A little background on the nematode: in 2017, thousands of the nematodes were isolated from symptomatic beech leaves by David McCain at the Ohio Department of Agriculture. These were sent to nematologist Lynn Carta at the USDA Agricultural Research Service in Beltsville, where they're identified as the first recorded North American population of Little Anthus crenata, originally described in Japan. The North American population shows slightly different morphological and host range specificity compared to the Japanese population, thereby distinguishing its own subspecies, Little Anthus crenata mccannii. Due to the negative consequences of this disease, this nematode is currently considered one of the top 10 most important plant parasitic nematodes in the United States. Although the origin of LCM is unresolved, the fast dispersal of this nematode, along with an apparent lack of resistance in native established forests, suggests this nematode was recently introduced into the North America. LCM appears to be thriving within American beech forests across the Northeast. I know you already saw this map, but I wanted to point out a few things. We first found LCM in Ohio County, or in Ohio and Lake County in 2012, and it quickly moved across the Northeast. You can see this map was made in 2020, but you can see how the disease started, and then we had these outliers far away from the original infestation. And as I move forward, you can see you had these jumps into Maine. This kind of raised a question of some sort of vector was involved and how it was spreading. And as I move forward, you can see some of these gaps are being filled in. So myself and some other researchers have been examining several different dispersal mechanisms. Short distance dispersal of LCM within a stand is probably due to rain or wind-blown rain. As these nematodes require a film of water for transmission, but it seemed like a vector was involved in the long distance dispersal. There's a number of factors leading to think that it could be vectored by birds. We've also been looking at insect vectors and the movement of nursery stock. So why do we think that birds could be a vector? It wouldn't be the first time that birds were associated with the spread of a disease. The potato cyst nematode has been shown to survive the guts of several bird species, including European starlings and the white-throated sparrows, and were recovered 24 to 48 hours after feeding. LCM has also been shown to survive in acidic environments, such as the bird's stomach. In 2020, Lynn Carta conducted some experience testing acid tolerance and found that 75% of LCM nematodes survived a pH of 2, and 10% survived a pH of 1.7 after 24 hours. These pH levels align with the acidity of many bird species' stomachs. Nematologists at the USDA ARS also detected a nonspecific associated with uncommon saprophytic mite. So you can see in this photo a picture of a predatory mite with nematodes, adults, and eggs attached. Mites or other feeding insects on American beech could have nematodes on them and be picked up on the bird's feathers, beak, and feet. Finally, LCM nematodes and eggs are more concentrated in American beech buds in the wintertime, which could be a food source for birds. While resources are scarce, birds may ingest LCM nematodes through bud and excrete viable nematodes into unaffected beech areas. So we wanted to look at both internal and external spread mechanisms. From internal, we hypothesized birds may be feeding on beech buds and spreading it in their feces. We also want to test for external presence of LCM nematodes, either attached to mites or directly attached to birds. As I said, birds may acquire LCM as they're flying through a beech forest or when they land on a twig infested with LCM. So for this test, we collected feather samples from the breast, crown, and vent of the bird. We also dusted them with diatomaceous earth and collected dust to look for nematodes associated with mites. Our studies took place at Holden Arboretum and the Allegheny National Forest. We stocked bird feeders with beech buds collected with beech leaf disease infected buds. So we found the buds in infected stands and we tested a subspecies of them to see if the nematode was present, put them in the bird feeders, and then we put mist nets around the bird feeders and caught birds, and we put them in these little paper bags for a really short holding period, 5 to 15 minutes. And I just want to be clear, we were nice to them. I really like birds. But we found several species of birds are willing to take beech buds from the feeders, including red-bellied woodpecker, downy woodpecker, song sparrow, black-capped chickadee, tuft titmouse, and American goldfinch. So the one issue with the way that this experiment was laid out is we weren't sure if the birds had time to ingest the birds and digest them between catching them. So we later tried this cage experiment. So we put the birds in a cage for a brief hold, and then we would either manually feed them buds or allow them to eat from a container, or we would pipette nematodes directly in their mouths. Again, we were really nice to them. If they seemed stressed out, we'd let them go. So here's a nematode isolated from the feces of a tuft-tip mouse we lovingly named Poopsie. She was really patient with her experiments, and she produced a lot of samples. She was a star patient. In both cases, we collected bird feathers, dust samples, and fecal samples from all the birds and tested them with molecular methods using Beech, like LCM specific primers. And we also looked for the nematodes under a microscope. In total, we collected 156 birds from 19 species. We successfully isolated and identified LCM DNA from fecal samples. We did get a higher recovery rate from the cage method, as you might expect, because we were kind of forcing them to eat it. 17% of fecal samples from the cage birds were positive, including a tuft-tip mouse, downy woodpecker, and white-breasted nuthatch. Only 3% of the birds caught passively had positive DNA, but I still think that's remarkable because it's showing that the nematodes are passing through their guts. We also had positive DNA from both feather samples and dusting. 10% of diatomaceous earth samples were positive, including those from a tuft-tip mouse, downy woodpecker, and dark-eyed junco. And feather samples showed a 7% positive rate for LCM DNA. We did observe adults and juveniles in fecal samples underneath the microscope. However, they weren't moving, so it was difficult to determine the viability of the observed life stage. So our results are promising. They show that the birds could possibly spread LCM, but we want to continue this project to confirm that the nematodes are viable before confirming that the transmission is possible. So I also wanted to talk about this project. I partner with Mihail Cantor and Sharon Reed on an international programs funded project that was accepted. The objectives of the proposal were to look at additional insect and plant vectors such as pollen, nuts, and wind. We also wanted to test the viability of transmission and likelihood of survival within the gastrointestinal tracts of some of those insects. So Mihail has shown in other experiments that LCM nematodes can pass through the guts of a caterpillar, such as this tussock moth, and survive. So we wanted to expand on this work further, potentially feeding those caterpillars to birds. Another aspect of this project would look at potential of how international trade between the US and Canada may have impacted spread, such as the movement of nursery stock and wood material. So I want to be clear that this is unpublished data that belongs to Mihail Cantor's lab. So please do not share this without his permission. But our partners at Mihail and his PhD student conducted a funnel test to see if LCM nematodes could be spread airily. So to test for this, they put out funnel stands at varying distances from infected beech stands. Each stand consisted of 3 different plastic funnels fixed to the stand, and then little vials were placed underneath the funnels to collect liquid. Over 300 samples were collected every other day for 2 months over 2023. The vials were brought back to the lab, and then they counted the nematodes, which was really labor intensive. They did find many nematodes in the funnels, showing that it can be spread airily, which hasn't been shown before and is very interesting. I know I'm going fast because I'm short on time. So I just wanted to say in summary, they found that larger tree diameters and closer proximity to the funnels had much higher nematode counts, just as you would expect. Additionally, they found that higher temperature, humidity, and wind speed was correlated with higher nematode counts, with the exception of super high wind speed and high precipitation combination. So we know that LCM can travel through the wind airily, but we don't know how far. So Mihail is going to repeat some of these tests this year with funnels further away. It's very exciting work. I'm also collaborating with Paul Vera and Bob Mara and Cameron McIntyre on a project that was funded by international programs to look at the origin of LCM. For this project, we want to look at the population structure of LCM microsatellites to determine the extent of any clonal population structures and to estimate the number of introductions into North America. So we're also planning a trip to Japan to look for LCM on Fagus species planted and native to Japan to ascertain if nematode species and subspecies present, as well as document the current distribution of within Japan. So I did contact APHIS in Japan, and there's no records of beech seedling movement into the United States. It's a regulated species, but unless it was moved illegally, it wasn't probably brought on beech stock. However, LCM could arrive on a different tree species or on infected seed, perhaps. So I know that was super fast, but I was already over my time, so I had to go fast. But if you have any more questions, that's my information. Thanks.
All right. Thank you, Danielle. That's great. Great information. We'll go on to our next speaker, Brady Holko from Davey Tree Lab.
Awesome. Okay. Hopefully you all can see me, but my name is Brady Holko. I am a botanist and laboratory technician. I've worked with Davey for about 4 years now, and about 3 of those years I've been working on this beech leaf disease study. And I believe that we're actually, yeah, let's start back here. So let's begin. So the speakers before me did a really excellent job of kind of giving you a little bit of the symptomology and biology of the disease, but I really wanted to talk about what we're doing to actually manage this disease. So just a little bit of history of our management research here at Davey. The first detection of beech leaf disease was in Lake County, Ohio, which is actually just 2 counties north of where the Davey headquarters and where my lab is located. In 2014, it was detected in the Cleveland Metro Parks, which is just a county adjacent to Lake County. The Cleveland Metro Parks began their annual beech and BLD surveys in 2016, and in 2017, the Cleveland Metro Parks and Davey initiated our management research. So we've been doing this for about 6 years now. And the product that we've been focusing on in our research is a product called polyphosphite 30, which I will talk about a little bit more, but in the future here. But the main things that we were focusing on were the effects of this polyphosphite 30 soil applications on both tree health and the nematode population density in the leaves. So this little photo on the very bottom here are photos of the nematode that I took under a dissecting microscope during our research. So a little bit about the actual product that we've been using. Polyphosphite 30 is a product from the Plant Food Co. It is a phosphonate, which is a phosphite product. It is a 0027 potash fertilizer or potassium polyphosphite. It is not a registered pesticide, although it does have the same active ingredients as Reliant and Agrifos fungicides. Since this disease is strongly correlated with a nematode, the fungicide won't really have any effect on it. And curiously enough, we don't really know a whole lot about the mode of action of this phosphonate product, but we do believe that it is a natural defense activator. So when we started this this whole study, we had 40 trees, 20 trees were treated and 20 were untreated controls. The trees were between 2 and 4 inches in DBH, diameter at breast height. They were saplings with mild symptoms at the beginning of the study. We trenched the several root grafts at the very beginning of the study. We applied this product at a rate of 2 ounces of polyphosphite 30 and 14 ounces of water per inch DBH. We had 2 injection points per inch DBH, and we did 2 applications per year, one in the beginning of May or June, and the second in August. So we're coming up on our first application date for the year. The trees were then, after the very last application of the year, evaluated by the Cleveland Metro Parks. And like I said, it was after that final application that we did. And they really looked at the canopy cover and branch dieback. So they had their own kind of scale on how they were rating these trees. So like I said, they looked at canopy cover between 0 and 100%, and they also looked at twig and branch dieback, more so the fine twig dieback and the main branch dieback. And they got really interesting results. We found that phosphite treatments maintain canopy health and decrease branch dieback. So in the chart kind of highlighted in green, you can see that our treated trees had a much higher percentage of canopy cover than our control trees. And the chart highlighted in blue, you can see that our treated trees had a very low percentage of dead fine branches compared to our control trees. And that was really evident in our research plots. If you were to go through the Cleveland Metro Parks and look at our research plots, this is what you would see. It's pretty obvious. Our treated trees have very full canopies. There's very little banding or symptoms. And our control trees are very sparse. The canopy is very thin. There's really heavy symptoms on the leaves, and the canopies are really dying back. So after our final application and the Cleveland Metro Parks went out and did their own ratings, that's when people in our lab would go out and collect the leaves to do our foliar nematode sampling. So the leaves were collected in early September after the Metro Parks went out and did their surveys. The leaves chosen were representative of symptoms displayed by the trees. So we had a scale that was between 0 and 6. And 6 was complete mortality, 0 was asymptomatic. So we chose leaves that were representative of the entire tree that we saw. So in 2021, we did 1 disc per leaf punch from a standardized location. So here we, where that circle is, that's where we took our little hole punch. So we took a cork borer and we just punched these little holes in the leaves. And in 2022, we did 1 disc per leaf punch from the standardized location where that red ring is, and another randomized location. And then the nematodes were counted in the Davy Diagnostic Lab. So when I say in 2022, we also did a randomized location, is we separated these leaves into these different sectors, and we labeled them 1 through 6, and we randomly chose a number where we would punch another hole at. And we got really interesting data. So in 2021, you can see that the nematodes that we counted in the leaves from the trees treated with polyphosphate 30 were significantly lower than those that were untreated. And we saw the same exact thing in 2022. And we also found that those random versus standardized locations don't really matter. So in 2023, we changed things up a little bit. So in 2023, half the previously treated trees were left untreated, and half the previously untreated trees were treated. So it looks something like this. Instead of 2 treatment groups, we ended up having 4 treatment groups with 10 replicates. So the first group, we had trees that were never treated. In our second group, we had trees that were treated every year between 2017 and 2023. In our third group, we had trees that were treated every year but 2023. And we had trees that were treated only in 2023. And we got some really interesting results when we went and counted the nematodes. So the nematodes counted from leaves of trees that were never treated were the highest. And the trees that were treated every year were absolutely the lowest. There was very little nematodes, if any at all. Trees that were treated every year but 2023, when we just started the treatments this year, had a very significant decrease in the amount of nematodes that we counted. And trees that were treated only in 2023 also showed a decrease in those nematodes that we counted. So we got, as a little summary here, as per the Cleveland Metro Park surveys, the treated trees had a greater canopy cover than our untreated control trees. They had a greater percentage of healthy leaves, and also a lower percentage of dead main and fine branches. And like I said, if you went through our research plots, you would see a pretty clear difference between treated and untreated without knowing that these trees were treated versus untreated. The polyphosphate 30 treatments are very effective at decreasing nematode density in the foliage. The population numbers are greater in areas of the leaf where symptoms are heavier. That was just something that we had noticed. The treated trees, untreated for a year, still had low nematode populations a year after treatment. So if we had stopped treatment just for a year, those same trees had very low nematode counts. And also untreated trees treated for only a year had nematode populations smaller than in previous years. So this whole study is fantastic. We've gotten a lot of really interesting results, but there's still some knowledge gaps that we have. We don't know everything about this product that we're using. So we don't know the mode of action. We don't know if the effects are direct. We don't know if it's having any direct influence on the health of the nematode itself. We don't know if it's absolutely just killing these nematodes. We're not really sure how it's working, but we do know that polyphosphate 30 and phosphonates in general do have a tendency to boost the immunity or the immune system of these trees. So it may be host mediated. We don't know the optimal rates or timing. The rate that we're applying this product seems to be pretty ideal. We are getting good results with it. We don't know if the effective rates scale with tree size, and you might be asking, well, if you did all of this research on small trees, what about large trees? And I will talk about that in a little bit. We don't know if it has curative effects. We don't know if it can impact trees or make them recover following treatment. We still don't have any data on that. And we also don't know if there's non-target impacts. I did receive a question a while ago saying if because this product is a fertilizer, if there would be any salts left over in the soil. And we did run some soil tests last year, and we found that there was no increase in those soluble salts in the soil. So what's next? What's next on our agenda? We want to see if polyphosphate 30 will be as effective on large trees as it is our saplings. So we started that in 2021. There's me doing a soil drench in the Cleveland Metro Parks. So yeah, with that, I'll open it to questions. All the questions can be entered into that little Q&A spot down at the very bottom. And if you have any questions, please feel free to reach out to me. All of my contact information is on the slide. Thank you.
Excellent. Very good job, Brady. We're gonna move on to the next part of our webinar with Dr. Sarah Tangren with the Metropolitan Council of Governments.
All right. So hello, everybody. That was, I've really enjoyed the presentation so far. Mine is about how you can report beech leaf disease if you see it. And the first presenter mentioned that Invader Detectives is, iNaturalist is a very good way to do that. And so we already have an early detection system set up for the National Capital Region. But generally speaking, you can use iNaturalist anywhere in the world. So this is a fantastic approach that you can use. For our own program, the Invader Detectives is hosted by iNaturalist, and that just very quickly, the area that you see in red here is red because of all of the 10,000 observations that citizen scientists have made of early detection species that the National Capital Prism is currently tracking. And for those of you that have used iNaturalist, you will know that when you make an observation on a smartphone in the field, that observation literally shows up on the World Wide Web within about 2 minutes. So it's an absolutely amazing system. It's incredibly easy to do. You just get your smartphone, you upload the free iNaturalist app. You look for early detection species. Invader Detectives is tracking about 100 species right now, one of which is beech leaf disease. But as long as someone is monitoring, and probably all of your counties at this point, the foresters are monitoring iNaturalist for beech leaf disease. So as long as someone is monitoring, your report will be available to them almost instantly. So to do that, you just get your smartphone, you hit that observe button at the bottom. The next window that comes up on your smartphone will show you a thumbnail of the photograph you just took. You can decide whether you want to keep that photograph or hit the plus if you want to add more photos. More photos are literally like always good. I think in the field it will let you take 4 photos or 6 photos. And so in addition to good photographs of the leaves, you could take a photograph that would be helpful to your local foresters, trying, if they wanted to try and find the area where you took the photograph. So like if you took pictures of the leaves for your first photo and then stand back and pictures of trees, pictures that include a path or a river or anything else that they might see, just in case your coordinates don't turn out to be terribly accurate. There's a little field here for notes. And very few iNaturalist observers leave notes. So hey, even you experienced iNatters out there, listen up. Please leave us some notes. So in this case, you might leave a note like, how extensive do you think the infestation might be? Did you see like acres of trees, or did you just happen to notice one tree, or were you only in the area briefly? And I'm hoping that maybe some of our expert panelists will give me some advice. If I see a tree with beech leaf disease, or probably has beech leaf disease, has a very distinctive pattern on the leaf, do you want me to go hiking around the area to see how large that infestation is, or should I just get out? Because I might become a vector myself. But anyhow, remember to leave us a note. And then this wild question here, it doesn't apply to beech leaf disease. So just don't even worry about that. This is just a screenshot of what your observation would look like. You'll be able to go to the website and look at this and correspond with other people. And we do have a listserv. I can't type in the chat, but Joanne, I don't know if you can type in the chat. Maybe you can leave folks my email address, stangren@mwcog.org, and put listserv in the subject line, and we'll add you to the listserv. And that's it.
We did put that information in the chat box. So the
Thank
link
you.
to iNaturalist and your email and instruction is in the chat.
Okay, thank you.
All right. Thank you, Sarah. And yeah, I'm a big proponent of iNaturalist myself. It's a great way to identify things if you don't know what it is. And yeah, just if you're nerdy about that kind of stuff, it's a great tool. And Sarah's correct. My office does mine the data from iNaturalist, and we get alerts when there are certain pests of our interest that are located in our county. We're like, oh, okay, we got to check this out. So thank you. So we have a question in the Q&A from Ryan. What other macronutrients were tested, if any? So I'm assuming this question is for Brady.
I'm not entirely sure, what you mean by that question. Do you mean like when we did our soil testing, or, if we tested any other kind of like nitrogen on these trees?
Yeah, I don't know. I guess, to the best of your ability from the short, yeah, in the study, I guess, yeah, we're macronutrients studied or tested.
Yeah, so, we had only used polyphosphate 30 in this study. We haven't tested any other product.
Very good, very good. So I had a question for Danny, my own personal. Oh, sorry. So I had, Ryan gave a follow-up, PP30 being phosphorus heavy, correct?
Yes, it is a phosphonate product, and I don't believe that there was any, and I apologize, I would have to look back at our soil reports, but I don't believe that there was any concerning amounts of any other macronutrients found in the soil.
Excellent. Thank you. I had my own personal question. Sorry, Danny, to put you on the spot. So there are other pests associated with beech, like beech bark disease. And so I don't know if you've come across instances where beech leaf disease has been found in the same area as beech bark disease. And I guess I'm assuming the mortality rate is accelerated in those cases.
Yeah, it's definitely found in concurring locations. And one of the things we do have, I didn't talk about this, but we do have long-term monitoring plots throughout Region 9. And one of the things we're looking at is beech bark disease and beech leaf disease occurrence and how they might interact and increase disease severity, and how beech bark disease might predispose beech to beech leaf disease infection. So that is, as the name implies, a long-term project. So we are kind of in the process of starting analyzing that data, because we do have a few years worth of data now. It does make sense that beech bark disease would predispose beech to infection with beech leaf disease, just because it stresses it out. And it's kind of a double whammy.
So we have a question from Edward. He asked, is there any material we can spread under our beech trees to potentially limit infestation? He gives an example of pot ash.
I'm assuming that's for Brady.
Yeah.
As far as to potentially limit infestations, I'm not quite sure if there is a product for that. But and like I said, I'm not really quite sure if potash would be effective either. As far as I know, we don't really have any way to potentially limit those infestations of the nematode.
Yeah. Yeah, one observation I found interesting is that with the infestations, the way that they're found don't necessarily like geographically make sense, where like you might find one in one state and then find another infestation 1 or 2 states over. So it's I find those patterns very interesting, just because it may just be a matter of people being aware of what to look for more than necessarily.
I agree. It is interesting. And there's a lot of factors at play. People could be looking for it more, and that's why you're seeing it more, but there could be other biological factors occurring.
Yeah, like the first infestation in Virginia was found at Prince William Forest Park at a national park just because a nematologist happened to be hiking there.
Mm,
Knew what to look for.
Right.
So, so we have a question from Susan. She says that, and I guess this would apply to anyone who has property that would be interested in being part of any research, but she has 30 acres in Winchester and would be more than happy to have people conducting studies on her property. And so I guess for anyone who's interested in loaning their property out for research, I guess, yeah, Danny or Brady, if there was interest, could they contact you?
sure.
I am unfortunately at the mercy of Davy, so I'm not the one that makes that, but yeah, I think that would be awesome.
Excellent. Let's see. Oh, there's another question from Candace. Are phosphite treatments only done as a soil drench? Are beech trees ever sprayed with phosphite?
Yeah, so in our study, we did soil injections for our saplings and soil drenches for larger trees, just because as far as applications go, it just made the most sense for the quantity that we're using on the trees. But we have never sprayed the trees with the phosphite.
And Brady, I have a follow-up question. So are there any concerns about like the use of this treatment and like water quality, like in the area, like if there's just use under the under the label if whether it's restricted to any area, like not in areas where it's like near bodies of water or anything like that.
Sure, and that's a really good question. And unfortunately, it really does fall into stuff that we haven't looked at quite yet. We don't know the environmental side of things, but as far as the label goes, I don't believe that anything is on that because it is not labeled as a pesticide.
Okay, thank you. All right, so we have we have a question here. So if we're in a forest and we see beech leaf disease symptoms, should we scout around for a while or should we snap a quick picture and get out? Are we in danger of becoming a vector?
I worry about this every day. Like I spray my shoes with alcohol, but I'm like, what if it's on my backpack? It's just a big concern of mine.
Yeah. So I guess that is a protocol though, right? Is that like an official protocol if you were to walk or if you work in an area that has beech leaf disease? Is that
I
like,
did a little mini study in my lab where I put some nematodes on a slide and then I sprayed them either with a bleach solution or with a alcohol solution or like something like a household cleaner that was either or. And I found that the alcohol solution seemed to kill the nematodes more effectively on my slide. But like whether or not that's going to work on your shoes or not, I'm not sure, but that's the best I have. Like, that's what I'm doing right now because it makes me feel better.
And I will second that whenever we're out doing our treatments. I will spray my shoes too, because like Danielle, I am also really terrified of accidentally spreading it. So
Okay, so I guess for the people who are out there taking photographs for iNaturalist, because there's some uncertainty, if you see it, maybe you're on a hiking trail or something, just kind of stay on the trail, take a good enough picture, and then don't go hiking through the forest. Taking more pictures. Until we know more. You guys are getting information so fast.
Like I've talked to my collaborators about this, and if it is spreading airily, it's really going to be hard to control because it's going to be in your hair and your clothing.
Right.
I do think about that, and I do go home and take a shower and wash my clothes when I go home to make sure before I go to another beach area. And I encourage other people to do so, but I hope that's effective, but I don't know.
Yeah, no, the same here. Because I work in the field and then I come home and I have these mature beech trees in my front yard. So
Oh, yeah.
yeah. All right. Well, I think that is it. That concludes the question and answer portion. Thank you, everyone, for speaking. I think, this is a great job. We have this webinar is being recorded. And so there will be a link eventually to the recording on YouTube, and Extension and Fairfax County will be sharing this. So yeah, when that becomes available, I hope all the attendees go ahead and share that link with everyone that they know and spread the word about beech leaf disease. This is a pretty unknown pest that's occurring in our landscape now. And so the more that we spread the word, the more people are aware and can prepare themselves for when eventually this pest either knowing that it's already in their community or preparing before it gets there. So thank you, everyone, and have a great evening.
Bye.
Good night, everybody.